happy to be here with you all. Actually, in this live, we're going to discuss about how the diversity of organisms on this planet. So, how we can study this this much diversity life on the land on this planet. Okay, so here I'm going to say a few words about how can we our ancestors brings their ideology to analysis and understanding of the different organisms and their life process. Okay, so recently I studied in one topic, that is diversity of life. Diversity of life, in which what will happen, so there is a lot of living organisms, many living organisms, different kinds, okay, different species. It's very difficult to study all these living organisms at a time. So our people, our scientific people decided to identify characterization. AP Decandle, who coined the term taxonomy. This guy first time used the word taxonomy. Okay, so what we will do in taxonomy: identification, nomenclature, identification. Then after we characterize that organism. We study the ethology and all the things, and morphology, where it will fit, in which group it will fit. We study that one. Then after nomenclature and classification. Okay, these things we use. Okay, then after mostly we people, we know zoology. So what we do, what is the uses of this one? We study this animal world with scientific principles. Okay, and we use different types of terminologies in the diversity of life. Like morphology we need, then after it has two things: internal morphology and external morphology. And in internal morphology you need to know of anatomy, histology, and organism ecology, ethology. Then after it's genetics, evolution, developmental biology, embryology. Then after all these things. Okay, for example, let me see what is morphology. It has two things: internal morphology and external morphology. What we need to study in internal morphology? Anatomy. Anatomy means internal arrangement of different organs. And histology, the term Mary Francois Xavier Bichat, who used he was the father of modern histology, Mary Francois Xavier Bichat. Okay, in histology we study the microscopic structure of different tissues, whatever the organ it is in the body. But what we do, we take like biopsy, we take a small tissue from that one and we study at microscopic level. It is called histology, nothing but microanatomy. Okay, microanatomy. Then after what we need to study of the things, living organism, it's ecology. Ecology means you took a organism, a living organism. Yeah, we have to study how it is interacting with abiotic factors and other living organisms. Okay, we have to study that one. That is called ecology. Ethology means it's animal behavior. Okay, these are all things. Then after heredity. Heredity means like, for example, we know that every living organism reproduce. Mostly living organisms are reproducible. Some are like countable to some living organisms will be infertile, but mostly organisms are those organisms are fertile. It has the capability to reproduce by different methods. In this, what happened? if we come to discuss about the sexual method of reproduction, organisms share their genetic material in the form of haploid gametes. Okay, haploid gametes. These haploid gametes consists haploid set of chromosomes. Okay, gametes consist haploid set of chromosomes. So, by this manner, organisms provide genetic information to the next generation. Okay. So, what type of similar characters transfer from parental organisms to their next generation is called heredity. What are the differences present from parental generation to offspring generation? Is called variations. The study of variations we call what we call genetics. Okay, what we call genetics. Okay, so firstly, many people we have also seen different. We will see every day lot of organisms. Okay, but we never care of them. We don't want, like, am I right? So, but our ancestors and this animal world have to study. Okay, we have to study this animal world. There is a, because by understanding this animal world, we can get most of information. How can we survive on this planet? How can we survive on this planet? Okay, so our ancestors, firstly, what they've start, they don't know. They used to roam here and there, different places for their food, and use it to fight with different animals. Okay, then after they've used to have to fill to fulfill their stomach, they used to have different fruits what are they available. So while this journey, our ancestors, firstly, for example, if they eat one fruit, what happened? If they eat one fruit, if they feel uncomfortable, like if they feel very abnormal, if they feel abnormal conditions after eating that fruit, they used to remember that we should not eat this fruit, like that. Okay, so some people used to die by eating different fruits in the past. So uh, then our ancestors did, by growing, our ancestors, what they did, they used to make symbols on the different rocks, on the different rocks, to find that plants and fruits which is useful to the humans. And also some of their symbols used to indicate the plants which is harmful to have, which is harmful to have. Okay, like that, we there is a particular terminology to that one. That means there is no literatures. Our ancestors used to keep all this data in the form of pictures, all the data in the form of pictures. Okay, and also this is a plant world, and also to the animal world also same, in the form of pictures also, only they use it to interpret it to the other generations. Okay, so by first it is in pictures, then after try to understand the world and plant world. Okay, animal world. You see, animal world is so big, so big. Plant world is so big, but it's very difficult to study each organism. Okay, so our ancestors come to one point, and by the classification of these organisms groups into groups, all similar organisms will be in one group. By studying one organism from that group will give complete mostly information of that all animals present in that group. Okay, by this one it's become easy to study the different organisms. Okay, then after, by coming there is a taxonomy, but what we call this classification we call as taxonomy. I will bring you what topic. one presently acceptable taxonomy, like Carl Lewis, and they used the three domain classifications: bacteria, archaea, and eukarya. Three domain classification. In bacteria, those prokaryotes which are similar, those single cell organisms which are needed mostly needed to, some are prokaryotes of bacteria and some are little like needed to animal world, eukaryotic domain. So they classified this as bacteria, archaea, and eukarya. Those really look like bacteria are under the bacteria, and which are needed to the eukarya are kept in the domain archaea. Okay. Then after, if you see the eukaryotic, this is the animal world. This is called as kingdom Animalia. Okay, what we this kingdom Animalia. In kingdom Animalia, if you see, there is two subkingdoms. Yeah. What with that? Two subkingdoms. That is Parazoa and Metazoa. What is Parazoa? Parazoa is the multicellular animals, but no well-defined tissues in that one. Okay, and that means only exhibit cellular life organization. For example, you go at that Parazoa animals, just you beat with a small stick, then it's become two parts. If you beat again, it's become more and more parts because there is no well-defined tissues. So they easily break, they easily disintegrate from one another. Okay, if you see the Eumetazoa, these are also multicellular animals, but include this subkingdom Eumetazoa includes multicellular animals with well-defined tissues. Okay, we are then we where will we be? Where the humans will be? Parazoa or Eumetazoa? Exactly, Eumetazoa. Okay. Then after Eumetazoa again into distinguished into two grades, like diploblastic and triploblastic animals. What is diploblastic? What is triploblastic? Let's see. Diploblastic means the organs of an organism arranged from two layers, ectoderm and endoderm. In triploblastic, three germinal layers present: ectoderm, endoderm, and mesoderm. Okay. In diploblastic animals, if you see, only consists two germinal layers, that is ectoderm and endoderm. Let us see which phylums will include in this diploblastic grade. Okay, what that are? Porphyria and Cnidaria. Okay, Porphyria will come in the Parazoa. In this grade, in diploblastic animals, Cnidaria and Ctenophora. Cnidaria and Ctenophora. These are the diploblastic phylums. Okay, phylum Cnidaria and Ctenophora will include under the diploblastic animals. If you see, its symmetry will be radial. That means very slow motion animals are these. Okay. Okay. So if you come to triploblastic animals, it consists three germinal layers: ectoderm, endoderm, mesoderm. Okay. Here in this grade, ectoderm, endoderm, mesoderm, those organisms which are like belongs to triploblastic animals will exhibit bilateral symmetry. Bilateral symmetry. Okay. For example, let me see the divisions: protostomians and deuterostomians. Divisions. These divisions belong to the grade triploblastic animals. If you see the protostomians, what's that? Protostomians. What is protostomians? We know that like the embryos of protostomia is called mosaic embryos. Very well known that word, right? So here, why we are calling as some organisms as protostomians? It's mean the blastopore at the time in the embryology first develops into mouth. Okay, in deuterostomes The blastopore develops into first anus, later mouth. That is the difference. Why we are calling some organelles protostomians, some organelles as deuterostomians. If you see the protostomians, it's include subdivisions like acoelomata, pseudocoelomata, and cytocoelomata. What is acoelomata? Acoelomata means A means absent, coelo means cavity, atom means having. Okay. No, it means simply no cavity. Okay. That means if you see the body wall and visceral organs, there is no cavity between body wall and visceral organs. It is filled with some fluids. That means no space between body wall and visceral organs. Okay, here that means how it will form. Including visceral organs it will form. Including outside will be in from the mesoderm. The mesenchyme will release and form the fills the space between outer body wall and inner visceral organs. So here the perivascular space is filled with mesenchyme, which is secreted from the mesoderm. By this one, there is no cavity. Okay, that's why we call all the organs which are present in the acoelomates as acoelomate solid body planes. What we call it as solid body planes. Okay. Next one. If you come to the first, which phyla will exhibit this type of thing? Which one? Platyhelminthes. This is the first triploblastic phyla. Okay, all the organisms in the platyhelminthes will exhibit. What will exhibit? Solid body plan. Okay. Next one, pseudocoelomates. That means it looks in these if you take the pseudocoelomates phylum nematoda, okay, exhibit pseudocoelomata. Pseudocoelom. That means it's look like a coelom but not a real coelom. That's why we call as pseudo means false cavity. Okay, it's look like the real coelom but it's not, it's a true coelom. Okay, let us see how it happened. Okay, there is a space between outer body wall and inner visceral organs. Okay, but it is not lined by the mesoderm. It is not lined by the mesoderm. So that means it is a remnant of blastocyst. What is it? It is remnant of the blastocyst. That means at the time of embryology, blastocyst. Okay, remnant of the from this one is the space formed. Okay, but it's not a true coelom. Next one is tube within a tube organisms. Nematode will exhibit this pseudocoelom. Good example to that one. Okay, generally you people will see like elephantiasis. That means the people will suffer with the big legs, very big legs. By that, what is that? There is no that lumbricoid something. I forgot that name. Which is belongs to that elephantiasis caused by the organism that belongs to the phylum Nematoda. Okay. Next one is what is the next one? Cytocoelomates. If you come to cytocoelomates, the organisms belongs to this subdivision. Have the true coelom, like outer layer is lined by the mesoderm here split into outer somatic layer, inner splanchnic layer. Okay, this somatic layer binds to outer body wall and splanchnic layer binds to the visceral organ. The space is the cytocoelom. The space created by this is called as cytocoelomates. Okay, which phylum will come under this one? Annelida, phylum Annelida, Arthropoda, Mollusca. These three phylums will come under this cytocoelomates. Okay. Okay, what is that one? and arthropoda mollusca. If you next division is deuterostomes, here what will happen? Okay, next one is deuterostomes. Here in deuterostomes, if you see, already I discussed about what is the deuterostomes. This means the first the plastopod develops into the anus. Later, the mouth will form opposite to the anus position. Okay, here the coelom, it we call as enterocoelom. The organism which the embryos of the deuterostomes is called as regulatory embryos. The coelom, which consists of the enterocoelom, will include in the deuterostomes. What is enterocoelom? Here, enterocoelom is splitting of the from the pouches of archater. The mesoderm is the cavity is formed by the pouches of the archater. Okay, this is also lined by mesodermal layer. Okay, this is the thing. Then after like this, the animals are classified. Which phylums will include in this enterocoelomates? Echinodermata, hemichordata, chordata. All these phylum, all the organisms in these phylums will come under the enterocoelomates. Okay, all these phylums will come under the enterocoelomates. Right. For more details in coming days, I provide my phone number also. For if you have any doubt in these discussion, you can call. And also will provide my mail. You can mail to that. Okay, for better interaction between us. Thank you. I hope you feel comfortable and friendly manner. Be feel friendly to make a call or ask your doubts or mail. If I not respond in phone, you can mail me. Thank you. Have a nice day.